one is probably that they put in some cases, ended up putting in too many dopamine neurons because there was no way to determine how many they were putting in. There's no way to test that. There was a second trial, and it was initiated shortly after that. Again, a double-blind clinical trial, again with fetal neuron transplantation. 34 patients, and they found the same thing. This was done in Florida, and this was done in Colorado. They're completely separated from each other. They got the same result, which means it's pretty clear what the result is. And this book, which was written by Kurt Fried and published in 2002, is really an interesting story about the development of the clinical trial and the outcome of the clinical trial, and from his personal perspective. He's a good friend of mine, and I know this is a really amazingly brave thing he did to initiate this sort of trial, to try to find out whether this treatment could be justified or not. Okay, because there were these problems with fetal tissue grafts for Parkinson's disease, including the ethical issue, using aborted fetuses is ethically complex. And there are quality control issues. If in each case you would need, for each person who was transplanted, you need multiple fetal brains. So you'd have to dissect out a little bit of the brain from many fetuses, and sometimes it's hard to have enough fetuses at the same time to transplant to a patient. So they had to store some of them over time, and they lost some of their abilities. So it just was very, very hard to control. There was also a difference because of abortions are not done in order to do this procedure. There's a lot of difference in the viability rate of the fetal tissue. There are different numbers of cells in each fetus and different kinds of cells depending on exactly how it was dissected. So there was also a necessity for immunosuppression because the fetal tissue did not match the person who was getting the transplant. And then, most importantly, this uncontrollable outcome, which is dyskinesia, in half of the patients that were transplanted. So because of that, in 2003, in a rather informal way, scientists decided not to continue with any more clinical trials for fetal cell transplants until they figured out how to avoid this bad side effect, dyskinesia.
Did the dyskinesia go away after a while?
Only in some of the patients. Some patients had to be treated for essentially what's the opposite effect of Parkinson's disease, which is also very unpleasant. Okay, so it became obvious when human embryonic stem cells were first made in 1998 that these stem cells might be able to be used for cell therapy for Parkinson's disease. We know that these cells can be differentiated into dopamine neurons. We can produce them in large quantities. They can be produced under quality controlled conditions. We know how many cells there are. We know what cells there are. And we can use good manufacturing practice, which is required by the FDA for use of products that are used in humans. The dosage can be controlled. The delivery method can be optimized. If the cells used are similar to the patient, you don't need immunosuppression. And there's a backup plan. If dyskinesia results, there is a way of putting a stealth gene inside these cells when you grow them up so that you can kill them by somebody taking a drug. So you can actually, if it goes wrong, you can fix it.
Backup. The backup plan.
Oh, sorry.
No. We just said that.
A backup plan. Oh, yeah, because that's the major problem. Right. It would be better to not get treated at all than to get that problem. Yeah. Okay, so this summarizes the idea of this perfect match in which you can take a patient, skin punch biopsy, make skin fibroblasts, make the pluripotent cells, make them in culture into dopamine neurons, and then transplant them back to the patient. Okay, so this is what we plan to do. We have divided our plan into 2 phases with very specific milestones and very, very doable parts. I mean, the steps are very doable. We've done them before, and we know it'll work. So the first thing to do is to culture the skin biopsy, and we've chosen to work on 6 donors with Parkinson's disease. We have Melissa Hauser will be the advisor about which patients we use. We'll create a frozen bank of their dermal fibroblasts using quality control measures. We'll generate induced pluripotent stem cells, iPSCs, from their fibroblasts. And we generally use 3 clones for each person. The quality control and characterization of iPS cells is something we're very good at. We'll create a frozen iPS cell bank and then differentiate these cells into dopamine neurons. And then at this stage, we need to transfer those cells to the good manufacturing practice conditions in order to develop clinical grade transplantable cells. And all of this has been done, but not in this, for this particular disease, and not in this order. So at the end of our phase 1, after we've made the cells, our plan is to transfer them to a colleague at the Buck Institute, Dr. Jeanne. She has a funded project to use the City of Hope GMP facility to culture pluripotent stem cells under GMP conditions, specifically dopamine-producing neurons from embryonic stem cells. So we would join her effort and provide more cells, in this case the induced pluripotent stem cells. They'd be grown under exactly the same conditions, with the outcome of having transplant-ready neurons.